Hello everyone, and for this video we will be teaching about the lac operon and catabolite repression. Since the world is a complex place, bacteria often find themselves in environments that have a variety of substances. They need to respond to these in order to survive. Organisms often need to regulate many unrelated genes simultaneously in response to changes in the environment. One way that microbes respond to the environment is through global control systems. These are regulatory mechanisms that respond to environmental signals by regulating the expression of many different genes. One example of a global control system that we'll be talking about today is the lac operon. The lac operon responds to global controls in addition to their own controls. Today we'll be talking about how cells respond when you give them more than one sugar. The following topics will be covered in this video: catabolite repression and details about it, dioxic growth, key players that make catabolite repression work, and regulation procedure of the lac operon. To start, you may be wondering what the process is called in which bacteria deal with multiple nutrients. What exactly happens when bacteria are confronted with several different usable carbon sources? For example, E. coli may be able to use different sugars and needs to know whether to use these simultaneously or one at a time. There must be a way for bacteria to know how to respond. The answer to their problem is catabolite repression. The definition is as follows: a mechanism of global control that controls the use of carbon sources if more than one is present. Overall, catabolite repression is an important part of the global control system of various bacteria. It will be covered more throughout the video.
Now we will look at catabolic repression in bacteria. In the presence of multiple carbon sources, bacteria chooses the most favorable source to catabolize first. The synthesis of enzymes that catabolize unfavorable carbon sources are then repressed. This ensures that the organism uses the best carbon and energy source first. For example, catabolic repression can be observed in E. coli. Because glucose is the most favorable energy source, its presence results in the repression of other catabolizing enzymes. Catabolite repression is also known as global control. Along with preventing the expression of other catabolic operons, catabolite repression can also control the genes which synthesize flagella. Flagella is utilized by bacteria to move to carbon-rich environments. When in the abundance of a carbon source, catabolite repression prevents the formation of flagellum, saving the cell energy.
I'll explain the consequences of catabolite repression. One of the consequences of catabolite repression is that it may lead to two exponential growth phases, called dioxic growth. In other words, when two energy sources are available, like glucose and lactose, the cells will grow first on the better energy source, which is glucose. When glucose is exhausted, there will be a lag period until growth on lactose occurs. The synthesis of lac operon proteins, including the enzyme beta-galactosidase, is also subject to catabolite repression. When glucose is present, the lac operon is not expressed and lactose is not used. However, when glucose is depleted, catabolite repression is abolished. Thus, the lac operon is expressed and cells grow in lactose.
In this section, we will be discussing some key players of catabolite repression. One of the main key players of catabolite repression is an activator protein called the cyclic AMP receptor protein. A gene that encodes a catabolite repressible enzyme is expressed only if CRP binds to DNA in the promoter region. This allows RNA polymerase to bind to the promoter. CRP is an allosteric protein and binds to DNA only if it is first bound to a small molecule called cyclic adenosine monophosphate, also known as CAMP or cyclic AMP. Cyclic AMP is a key molecule in many metabolic control systems, both in prokaryotes and eukaryotes. Since it is derived from a nucleic acid precursor, it is a regulatory nucleotide. It is synthesized from ATP by an enzyme called adenylate cyclase. In the presence of glucose, the synthesis of cyclic AMP is inhibited, and cyclic AMP transportation out of the cell is stimulated. Also, with glucose present in the cell, cyclic AMP levels is lowered and CRP can no longer bind to DNA. Therefore, RNA polymerase fails to bind to the promoters of operons subject to catabolite repression. The direct cause of catabolite repression is a low level of cyclic AMP, and that is why cyclic AMP plays an important role in catabolite repression.
In this section, we will cover regulation of the lac operon. The first regulatory pathway involves the protein LAC-I. LAC-I naturally binds to their operator and represses the expression of the lac operon gene. The presence of an inducer will inhibit the LAC-I protein by binding to it and inhibiting it through conformational change. The inducer is generally a product of lactose and is only present in the presence of lactose. The second regulatory pathway involves the protein CAP. CAP is usually inactive in cells. CAP is activated through binding with cyclic AMP. An activated CAP can then bind to the promoter and initiate transcription of the LAC gene through recruitment of RNA polymerase. Cyclic AMP is generated through adenyl cyclase, which is usually inhibited by glucose. The absence of glucose will allow production of cyclic AMP and activation of transcription in lac operon. It is important to note that the repression of LAC-I as well as the activation of CAP must both occur for transcription of the lac operon to proceed.